Hello, Namaskar and welcome to Micro Mahasutra by Dr. 𝗵𝗵𝗴𝗺 and I am your microbiology faculty. Today we are going to discuss the General Microbiology Session 3. In this session we are going to discuss about the bacterial toxins. So bacteria also has toxicity in his life. Let's discuss about the toxicity of bacterial life, that is bacterial toxins. Okay, my dear friends, the topic of toxins has been divided into three parts. One is endotoxins, exotoxins, and the difference between the endotoxin and the exotoxins. And in the end, we will discuss a topic of vital importance, that is mechanisms of exotoxins. Okay, so first of all, we will discuss about the endotoxin, the inner toxins, the inner ones. Okay, endotoxins. And I always say that I have a little narcissistic personality, and wherever suitable, I am so proud and happy to use my name as the mnemonic. Okay, 𝗵𝗵𝗴𝗿𝗺's mnemonics, 𝗵𝗵𝗴𝗺's, huh? So see here. So my name is 𝗵𝗵 and then 𝗵𝗺. So remember, 𝗵𝗺 is my surname, and that is something which we are going to use inside. So 𝗵𝗵 is on outside, 𝗵𝗺 is on inside. That means 𝗵𝗵 is the exo, 𝗵𝗺 is the endo. That means the endo is made by L for lipid, and P exo, P for protein. Okay, P 𝗵𝗵 exo protein, 𝗵𝗺 L endo. Okay, you got it? Great. So remember, that means the endotoxins for that matter are made up of lipid A. See here, 𝗵𝗺 endo lipid. Lipid A of lipopolysaccharide. And if you have not seen a previous session in which we discuss about the bacterial cell wall and the other appendages, I have discussed this in detail. So endotoxins are made up of lipid A, part of lipopolysaccharide. Okay, and it is the part of the cell wall of the gram-negative bacteria. So the gram-negative bacteria has a cell wall which has got an LPS layer. This LPS layer has got a lipid A, and this, because LPS layer, this is a whole layer that has got a lipid, which lipid A, responsible for production of toxin. And which toxin? Endotoxin. Which toxin? Endotoxin. Now what does this LPS layer does? In order to remember the functions or the pathophysiology of this LPS layer or of this lipopolysaccharide layer, we have got a mnemonic that says 𝗖𝗵𝗵𝗴𝗺. Okay, so you can just remember the lecture is at 8 p.m. 𝗖𝗵𝗵𝗴𝗺 8 p.m. Lecture at 8 p.m. What is L? L for lipopolysaccharide. जिनके हम लोग functions पढ़ रहे हैं. We are discussing the functions of lipopolysaccharide. E. E for endothelium. E for endothelium. C. C for complement system. C for complement system. At 8 p.m. What is P? P for platelets. P for platelet. What is M? M for mast cells. M for macrophage. M for mast cells. M for macrophages. So the mnemonic is lecture at 8 p.m. L for lipopolysaccharide. E for endothelium. C for complements. P for platelet. M for mast cell and macrophage. So the lipopolysaccharide is going to act on all these areas. L for lipo and all the different areas. When it acts upon the macrophage, it can cause what? It can cause the release of TNF-alpha and interleukin-1, which I've already told you that these are responsible for causing fever. TNF-alpha and interleukin-1. These are just the substances. And if you want to know the full form, macrophages are responsible for the release of TNF, that is tissue necrosis factor, which is responsible for the death of the tissue. Necrosis means death. So responsible for the death of the tissue. And IL means interleukin. Both of them are responsible for fever. Also, please understand that iron is responsible for the growth of bacteria, but lipopolysaccharides decreases the iron, and if the iron is decreased, then there will be inhibition of the bacterial growth. Okay, so this is the
a thing which you have to keep in mind. After that, it is responsible for acting upon the endothelium, acting upon the mast cells, acting upon the platelets. So what is happening when lipopolysaccharide acts upon the endothelium? So see, this is the blood vessel. In the blood vessels, these cells are nothing but the endothelium. This endothelium gets a split. When the lipopolysaccharide LPS acts on it, this is going to get split. And once it is getting split, the permeability is going to increase. So there will be increase in the vascular permeability because the endothelium is going to get split. Also, there will be mast cells which are going to release histamine. So the mast cell will release histamine, which is also responsible for increasing in the vascular permeability. And the platelets are also going to cause the increase in the vascular permeability. How? I'll tell you. But in the end, just remember, the vascular permeability is increased and that leads to hypotension. And whenever there is BP fall, this condition is ultimately going to cause shock. Why? Because BP is less. That means there will be not enough amount of blood which is reaching to the vital organ, and this is something which is called as shock. Shock means less tissue perfusion. That means the tissues are not getting bathed with the blood properly. That is shock. So basically, endothelium split, mast cell releasing histamine, and the platelets. All these three are going to cause increase in the vascular permeability. Okay, and ahead they are responsible for causing hypotension, and then they are responsible for causing the shock-like condition. Okay, and because this shock is happening because of bacteria, this is called as septic shock. Okay. Also, it leads to activation of clotting factors. So what happens whenever there is a split in the endothelium? It is going to stimulate the clotting factors. Whenever there is a split in the endothelium, it promotes the formation of the clotting factors. And once the clotting factors are being formed, it leads to the utilization of the platelets. It leads to the utilization of the platelets, and once the platelets are being utilized, it leads to DIC. It causes what? It causes DIC. Because the clotting factors are stimulated, the clotting pathway is activated and the platelets are being used unnecessarily. So this leads to intravascular coagulation, disseminated. Okay. So this is about the second function of the lipopolysaccharide. Third one is the complement activation. So in the complement activation, this leads to activation of two complements, C3A, C5A. 3 having double E is just like I. So I for inflammation. So C3A is responsible for inflammation, whereas C5A is responsible for chemotaxis. Taxis, chemotaxis, chemo, CHEMO, 5 letter, 5 words, C5A. So what is happening? See, C3A is responsible for inflammation, okay, and C5A is for the chemotaxis. So the neutrophil, here the WBC is there. WBC is going to the site where the fight is going to happen. WBC is going to the site where the fight is going to happen. Okay. So WBCs are going to the site with the help of what? Chemotaxis at the site of the fight to fight with the bacteria. Okay. So lipopolysaccharide itself is a trigger for the C5A complements, and then the C5A is responsible for the WBCs to go and to kill the bacteria. And one more effect which you have to just cram, that is hypoglycemia. That is what? That is hypoglycemia. Coming on to the next part, exotoxins. So I've told you endolipid, exoprotein. So these are made up of proteins, and generally proteins are heat-labile. Okay. So the proteins or the exotoxins are heat-labile. They are going to get killed by the heat. Okay. So have you heard the betadine gargles which we do? Like in the diphtheria also, we tell the patients to, even in pertussis, to do the betadine gargles, because the higher temperature, not the betadine, actually the hot water gargles, because the higher temperature is also going to kill the exotoxins. Okay, so heat-labile proteins, like that. And heat-labile proteins, P for proteins, remember this, I have told you 𝗵𝗺𝗺 endolipids, exoproteins, and P for two P's also. One is positivity. That means heat-labile proteins are present in gram positive as well as gram negative, whereas the lipid one, the endo one, are all only present in the gram negative ones, whereas these ones are present in the gram positive also. Okay, you got it? Good. Now, another P is PAS. What is PAS? PAS means one is the potency, antigenicity, and specificity. What is potency? Potency means that exotoxin is highly potent. Even a mere 30 gram of botulinum toxin, my dear friend, is sufficient to kill the whole mankind. Can you estimate the potency? 30 gram botulinum toxin can kill you, me, your father, mother, here, there, everyone. This is the potency level. Antigenicity. Exotoxins are antigenetic. That means anti means antibody. Genetic means generating. So they have got a capacity to generate the antibodies and to induce the immunity, my dear friends. And remember that they can be converted into toxoid because they are antibody generating. So why do we give vaccines? Vaccines we give to generate antibodies, so that when real infection comes, at the time of real infections, we have large antibodies to kill the real infection. So that's why it is called as, so you are actually you are putting an antigen, that is a toxin, but since it is a weak toxin, it is called as toxoid. So toxoid, like if you have listened to this, diphtheria toxoid, tetanus toxoid. So we are putting a small part of exotoxin in the human body, okay, sorry, in the vaccines, and then vaccines are injected into the human bodies because they have got, these exotoxins have got a property to generate the antibodies. It will generate the antibodies, and when the real infection will come, you will have sufficient antibodies that are going to kill the real infection. So we were discussing about the PAS, potency, antigenicity, and specificity. That means it is going to act specifically on some specific organs. For example, any specific cell, cytotoxic, any specific GIT, for example, enterotoxin, and if it is acting on CNS, it is called as neurotoxins. Okay. Coming on to the third part of this discussion, what is the difference between endotoxin and exotoxin? Exo means exit. They are going to get exit from a cell. That means they are going to get secreted. Exo means exit from a cell. They are going to get exit from a cell. That means they are going to get secreted. This is exotoxin. You got it? Great. So they are secreted, whereas endotoxin is going to be released by the cell lysis. The cell itself is going to get vanished, and then endotoxin is going to get released. That's why it is endo, it is inside. Exo means it is going to get released, exit, secreted. Okay. And I've told you exo from protein, endo from lipid, only in the gram negative, endo, both gram positive, gram negative, exo. Okay. And I've told you that these proteins are heat-labile, destroyed. These are heat stable, endo. Potency, PA, we have discussed, potency is high for exo. Antigenicity is also high. Toxoid formation is possible, like tetanus and diphtheria, I have discussed. Clinical features, you can easily remember. Fever because of interleukin 1 and TNF-alpha, we have discussed. Shock because of increase in vascular permeability. In fact, hypotension and DIC are also because of increase in vascular permeability. Then is the inflammation. That is because of C3A. THREE is just like I. So I is inflammation, and these are neurotoxic and cytotoxic. Coming on to the examples, very important. Please understand, this is a very important MCQ, that you have got a family called as Enterobacteriaceae. We'll discuss in detail. The Enterobacteriaceae has got E. coli, it has got Salmonella, it has got Shigella. Okay, so Enterobacteriaceae has got E. coli, Salmonella, Shigella, and in fact, for that matter, Pseudomonas you can also remember. So out of all of them, only the 𝗏𝗏 is having the exotoxin. So remember a girl name is 𝗏𝗏. 𝗏𝗏 wants to exit from her home. So 𝗏𝗏 toxin or 𝗏𝗏 toxin is a exotoxin. Rest all of them, E. coli, the Salmonella and Pseudomonas, they are endotoxins. You got it? Great. And diphtheria and tetanus we have already discussed in exotoxin. Only remember you have got BSC, that's the mnemonic for exotoxins. So 𝗏𝗏 is, exo. The the mnemonic, okay, so diphtheria you know, tetanus you know, as for 𝗏𝗏 you know, remaining are these. I can't make the mnemonic which you people desire, so that's why I am desiring the BSC. So if you can understand what I just said, you are grown up. If not, then welcome to the second year. Little toddlers. So BSC is the another mnemonic. B for botulinum, C for cholera, S for 𝗏𝗏, D diphtheria, T tetanus. So students, coming on to the last part, that is examples of exotoxin. Okay. In order to remember the examples of exotoxins and their mechanism, because this is very, very, very important, please don't miss out at least on those examples which I'm going to mark for you, because these are very important. The mnemonic is in the class 10th, up to class 10th, you have got a subject called as Social Sciences, SSC, and you have got a subject called as EVS, Environmental Sciences. SSC and EVS, both of them are very fond of using BCC, that is before Christ century, right? So I've taken this as a mnemonic to remember exotoxins. Now, since the EVS and SSC are exit from your curriculum, curriculum as in now you are not reading. So these are the examples of exotoxins and not the endotoxins. I know this might be a weird mnemonic, little weird, but then this is the perfect way to remember. SSC, EVS, use BC, before Christ century. Okay. So what are the examples? So SSC means, see, this is SSC, that is Staphylococcus aureus, Streptococcus pyogenes, Clostridium botulinum. SSC. EVS, E. coli, Vibrio cholera, Clostridium tetani. Uses BC century. B for Bacillus anthracis and C for Corynebacterium diphtheria. Okay, and one more S is there, Shigella. So see here, Corynebacterium diphtheria. What is the mechanism of action? So Corynebacterium diphtheria produces diphtherial toxins, and remember CDE. So CD means Corynebacterium diphtheria, E for elongation factor 2. So Corynebacterium diphtheria releasing the diphtherial toxin is going to inactivate the elongation factor 2. And once the elongation factor 2 is damaged, this is going to inhibit the protein synthesis by inactivating the elongation factor 2, decreasing the protein synthesis. Coming on to Bacillus anthracis. Remember, the mnemonic is ABC. So ABC means A for anthracis, B for bacillus, C for cyclic AMP. I've written ABC twice to tell you A for anthrax, bacillus, C is CA, cyclic AMP. Coming on to the next part, Shigella, name is Shigella dysentery, responsible for the release of 𝗏𝗏 toxins, is going to inhibit the protein synthesis. How to remember? PQRST. ST for Shigella toxin, 𝗏𝗏 toxin. P for protein synthesis inhibitor. Coming on to E. coli. E. coli is of two types. E. coli is of many types actually, but I am talking about ET E. coli. That means enterotoxigenic E. coli. Just remember the name, enterotoxigenic E. coli. It is responsible for synthesis of two toxins. One is going to be labile toxin, other is going to be stable toxin. Labile means it is heat-labile. So one toxin is sensitive to the heat, so it is going to get damaged when in exposure of heat. So this is called as heat-labile toxin. Other is called as heat-stable toxin. Remember the mnemonic Los Angeles. That means the labile toxin is going to act by cyclic AMP, whereas stable toxin is going to act by cyclic GMP. both cyclic AMP and cyclic GMP are going to increase the secretions from the gut, and because they are increasing the secretions from the gut, they are responsible for causing diarrhea, secretory diarrhea. Increasing the secretions, secretory diarrhea. Who? E. coli. Okay, you got it? And the next mnemonic is going to be Los Angeles and Las Vegas. LALV. What does that mean? Remember, just like the labile toxin was using cyclic AMP, similarly, that is Vibrio cholera, is going to use cyclic AMP and responsible for causing secretory diarrhea. Vibrio cholera का जो diarrhea है, Vibrio cholera's diarrhea is called as rice water stool. And I think the mnemonic is clear. LA and LV, that is Los Angeles and Las Vegas. Okay, coming on to the next part, that is Clostridium tetani. So normally what happens and what tetani does to your body? Please understand. So for example, normally in a normal individual, like I am sitting, you are sitting very close to me, right? So normally if our muscles are in some spasm, if the muscle is in spasm, there will be some inhibitory substances which are going to inhibit the muscle spasm. For example, GABA is an inhibitory neurotransmitter that is going to get released from my nerves and that is going to relax the muscle. I'm repeating. If your muscle is spasmic, if your muscle is in spasm, there will be release of some relaxing neurotransmitters or inhibitory neurotransmitters that are going to release the spasm, just like GABA, just like glycine. But Clostridium tetani releases Tetanospasmin. Clostridium tetani releases Tetanospasmin. That is going to block the release of GABA and glycine. And that's why the muscle in spasm remains in spasm for a longer period of time. So, Clostridium tetani is going to inhibit the release of these inhibitory substances and there will be no relaxation. There will be sustained spasm. There will be sustained spasm. So, it blocks the release of inhibitory neurotransmitters GABA and glycine. That leads to lockjaw. See, the jaw is locked. Lockjaw. Opisthotonus. That means the patient will be like this. That's a yogic posture also. The patient will be like that in that yogic posture. Sustained. Opisthotonus. Okay. Then comes Clostridium botulinum. That is responsible for release of botulinum toxin. All the girls out there who are studying with us, you're very, very much aware. Sir, do you know that we inject botulinum toxin so that there will be no wrinkles? Huh? You are aware. How? See, everybody knows acetylcholine, which is responsible for the action of the muscle. Okay. When there is wrinkling, that means your muscles are active and there is wrinkling. See? Can you see? Hey, don't look at my wrinkles. Anyways. So what we do, we inject the injection Botox, and once we inject the injection Botox, the muscle is stopping working. The muscles will be in paralysis, flaccid paralysis, and there will be no wrinkling. Okay. So remember the mnemonic ABC. A for acetylcholine, B for block, C for Clostridium, and this B for botulinum. So Clostridium, botulinum blocks the acetylcholine. That leads to flaccid paralysis and the skin is taut and the wrinkle free. Coming on to last example, Staphylococcus aureus, Staph aureus. Mnemonic is PQRST. S for staph, T for toxic shock syndrome toxin. The name of the toxin of Staph aureus is toxic shock syndrome toxin. And this staph has an habit to act on the superantigen. It has an habit to act on the superantigen and is responsible for shock. What is superantigen? I'll discuss in detail when I'll discuss Staphylococcus aureus. But as of now, remember, staph acts on superantigen and responsible for causing shock. Okay. And similarly, Streptococcus pyogenes, SS superantigen. Remember, with the help of mnemonic sepsis, E for the name of the toxin, erythrogenic toxin, Streptococcus pyogenes, E for erythrogenic toxin. It also acts with the help of superantigen. It also acts on superantigen. I just hope that my class was, actually it was full of toxins, but I hope that it was not toxic to you. It was easy to you. You have enjoyed it. Always the comment section is open for your feedback. Thank you so much for watching us. Please share your love, support, blessings, and some little positive feedbacks which keep us growing, going, and working harder for you. Thank you.